Hello everybody, this is Mr. Henson, and I'm here to talk to you today about Unit B2, which is cells. So we'll be running through stuff today quite quickly. Remember, because it's a video, you can always pause, rewind, and go back over things. Make sure you've got your revision notes out with you to complete as you go through the video, and this will help you when it comes time for revision. Remember, you can always access the videos on the website, so that's always going to be really handy for you. I also need to make sure that you guys are answering questions as you go through it. So please, when the video stops and our question is asked, do not skip on. Make sure you stop, answer the questions as fully as you can so that we can use it as a basis for discussion in the classroom. Okay, so without further ado, let's start looking at cells and what they are. So, where did this idea of cells come from? Well, Robert van Leeuwen, who was the first scientist or biologist ever to sort of conceive the idea of cells, was looking under a microscope at some cork tissue, okay, the cambium of cork, so it's sort of woody tissue. And he noticed that it was compartmentalized into these little squares here. And what he ended up doing was calling these cells because it reminded him of the cells in a monastery. Okay, so these reminded him of the cells in a monastery. Very bare, not much in them. It didn't look like at the time, and this is what he likened them to. Later on, of course, it wasn't until better technology came out where people started realizing there was actually a lot more in these cells. Okay, and that these cells were actually the basic units of life. Okay, now obviously with emerging technologies, it became much easier to identify cells and actually figure out that there were things inside them. And so this might be the sort of general image you might think of when you think about cells. Okay, you've got this, you've got the cell membrane, which is this boundary around the outside. You've got the purple blob in the middle, which is the nucleus. But some of these other images here might be, you might know less about, but it's important to realize that with technology, we can see cells now in lots of different kind of 2D and 3D projections. So this is an image of a scanning electron microscope looking at the bottom of a leaf. And you can see these. Now this is actually two cells. You can't see the definition, but these are guard cells. And this is a stomata in the bottom of a leaf. So each of those, maybe if you can imagine an imaginary line going down the center, you've got one guard cell here and another guard cell here. This is a cell that's used for that. Over on the top right-hand corner, okay, you've got another electron microscope image of white blood cells, or actually they're called phagocytes, which actually go around in your blood and they kind of hoover up or eat any kind of bacteria that they might find, okay, and we call these phagocytes. And again, this is obviously taken from outside, looking into the cell, looking at this sort of image. This is what we call a scanning electron microscope again, because it's just scanning the outside. These images here were taken with much more, still electron microscopy, okay, but they're called a transmission electron microscope. So on this one, look, we can see what's inside the cell and we can make out what's going on in each cell. So this one here is the nucleus. You can see here this individual chromosomes of this particular cell. Now these are from a root hair cell. So they're ones that are dividing where the DNA is dividing very quickly and making new cells very, very quickly because it's an area of growth, okay? Down here, you can see images of nerve cells. And here we've got, again, looking on the outside, we're actually looking at an adapted leaf that's been rolled up called marram grass. And actually, each of these things here is an individual plant cell. Down here, these are plant cells, and these green things that you can see inside are chloroplasts. So obviously we'll be discussing the major parts of the different types of cells today, but it's important to remember that cells come in lots of different shapes and forms, and that we'll be looking at quite a few different ones. So here is the basic image of the cell that you'll remember, okay? You've got your animal cell and your plant cells, okay? And the key similarities and differences. Now, you'll be actually doing some work looking at the different parts of the cells. And hopefully some of these parts you'll be able to identify, but some of them you probably won't, because they simply won't show up using our microscopes. So in any cell, nearly all cells, we have got a nucleus, which contains DNA in the form of chromosomes. So this is our nucleus in an animal cell, the nucleus in a plant cell. We've got cytoplasm, okay, which is the liquid part of the cell, which is where our chemical reactions like respiration and some parts of photosynthesis might happen in a plant cell. We've got a cell membrane, okay, which is what we call a selectively permeable membrane, because it lets some things in and controls what comes in and out. Not everything can get through it, not everything can get out of it, which is a very important feature. We've got mitochondria, these little black dots. We find them in both animal and plant cells, look. Okay, and these are the powerhouses of the cell. This is where the major parts of respiration occur, so where sugar is broken down to release energy in the form of something called ATP. Very important organelles. You don't get to know a huge amount about them in our GCSE, but you do need to know that they're the site of cellular respiration and that they produce energy in the form of ATP. Okay, so they're the similarities, the things that both animals and plant cells share. But plant cells have a few extra things. They've got the rigid cell wall, which is made of a chemical called cellulose, which provides it with a lot of support. They've got a vacuole in the center, which contains cell sap, which is a weak solution of sugar and salts in water. Okay, and it's very important in controlling how turgid the plant cell becomes. That word turgid will become a bit more familiar to you once we look at osmosis and diffusion. They've got green chloroplasts, which are these green areas here, which of course are for the absorption of sunlight energy for photosynthesis. So that's where plant cells are actually generating their own glucose through the process of photosynthesis. Some specialized cells, hopefully we might get a chance to look at these as well. Some specialized cells are red blood cells. Red blood cells have an amazing structure. They're round, round in shape, but they're biconcave, which means they're kind of compressed in the middle. You can see this compression on this side and a compression on this side. Okay, that's so the surface area of the red blood cell is increased so that they can carry more oxygen. Remember, red blood cells are how oxygen gets around our bodies, and so maximizing their surface area over which they can absorb that oxygen is very important. They don't have a nucleus, okay, so they can make more space to absorb oxygen. And they have a very specific chemical called hemoglobin, which is a protein which we find within the red blood cell, which binds to the oxygen. So they're really, really, really important. And just a different example of a cell. Now you can see this is a light microscope image. And you can see that biconcave shape, how it's represented in this image, is that it's paler. So it's the contrasting colors that we're seeing that show this shape, this paler color on the top where we're looking into. Okay, root hair cells are another very, very important type of cell. They grow off the roots. Now you can see this is a seed which has recently germinated, and the root or the radicle is growing downwards, and you can see these fine projections sticking out. Now these are made up of lots of root hair cells. So they're increasing the surface area over which they can absorb water. Remember, their primary function is to absorb water and also to absorb mineral salts like nitrates from the soil. They allow the movement of water by the process of osmosis. So the movement from high concentration of water in the soil to the range of low concentration in the root hairs, okay, through these root hair cells, okay, and into the epidermal cells, and then further on into the xylem. They have a large vacuole to absorb as much water, okay? And of course, they carry out the normal things that cells do. They carry out respiration because they need energy sometimes to actively transport things like water and minerals in. If they find that the soil is very, very dry, they can actively do that. Now I can see my battery is running low. There'll be a second part to this video, but I'm going to stop recording now. I hope you've enjoyed it. Please go back, rewind, make sure you answer the questions that are built into the video, and I hope you enjoyed listening.